Hello friends, welcome to SR Academy.
Welcome to SR Academy. Empowering minds, igniting curiosity. Your gateway to knowledge unleashed. Let's get started.
So today we are going to discuss about telomers and self-renewal. Under this topic we will be discussing about telomers and different models different researches which has been approaches have been focused on the telomers, and telomerase enzyme, how exactly it is helping to overcome the lagging strands, all these things that we will discuss in the this particular topic. So telomers, all of you know, so telomers are the repeated topics in the biotechnology subjects. So telomers are nothing but the region of DNA, okay, at the end of the linear eukaryotic chromosome. You have a chromosome, right? At the end of the eukaryotic chromosome, the region of whatever the region of DNA is there, that are required for the replication and as well as the stability of the chromosome. So that is called as telomers. So usually these telomers are nothing but the end part of the chromosomes. That's why they are called as telomers, right? In that end part of the chromosome, it's a region of DNA, right? So what are their these particular telomers functions? They will help. They should be there for replication and the stability of chromosome. So for that's why we are calling it as calling them as a telomers. So telomers are the region of DNA at the end of the linear eukaryotic chromosome, okay, that are required for the replication and the stability of the chromosome. So in order to have a chromosomal replication and stability, we should have telomers. Understanding? So that's the definition of telomers. So McClintock, she was one of the scientists. She recognized their special features. That means telomers' special features when she noticed that if two chromosomes were broken, imagine you have two chromosomes which are broken inside the cells. What happens? The two ends will be sticky, and end of one could attach to the end of other, or vice versa. Understanding? So imagine two chromosomes. Take a chromosome inside the cell. Now, if the ends are if they were broken, okay, one end of the one chromosome, that is chromosome A, can bind with the another end of the another chromosome, that is chromosome B. So here it can since the ends are sticky, when the chromosomes are broken, the ends are sticky. They will go and stick to the another chromosome. So the one end of the chromosome can combine with another chromosome, okay, another end of the chromosome, right, or vice versa. So however, she never observed that the attachment of the broken end to the end of the unbroken chromosome, suggesting that the end of the chromosome have unique features. So definitely she did the research, but here what she missed is she forgot, or she didn't observe the attachment of broken end to the end of the unbroken enzyme. Understanding? There she mentioned broken to broken. She missed studying from broken to unbroken chromosome. Understanding? The binding of broken chromosome to the unbroken chromosome, suggesting that the end of the chromosomes have unique features. So telomere sequences remain conserved throughout the vertebrates. They form caps that protect the chromosomes. These are all things we studied in chromosomes, from nucleases and other destabilizing influences, and they also prevent the end of the chromosomes from fusing one another. So the telomere sequences they remain conserved throughout the vertebrates. Okay, they form caps. These are some of the functions of the telomers. I'm listing it out. So they form caps that protect the chromosomes from nucleases and other stabilizing agents, and also they prevent the end of the chromosome, that is called as telomers, from diffusing with one to another. That means this telomere itself, they will protect, so that for the diffusion or fusing. So human telomers contain a sequence that is called as TTAGGG, which is repeated around 500 to 5000 times in humans. Okay, there are certain bacteria possesses telomers in their linear genetic material, which are of two types. So hairpin telomere in invertron telomers. So hairpin telomers. So why it is called as hairpin telomers? As the name itself it indicates, the structure it simply becomes like a hairpin kind of structure. That's why the name is hairpin telomere. So as its name implies, the telomers bend around the end of one strand of DNA, to the end of complementary strand of DNA. Understanding? Here we need to remember we are talking about DNA strands, not the one DNA or not different chromosomes or not individual DNAs. Understanding? So as its name implies, the telomere bend around one end of one DNA strand to the end of complementary DNA strand, right? So that's why it is called as hairpin telomere. Next, invertron telomere. Here this type acts to allow an overlap between the ends of the complementary DNA strands. So here there will be overlap there between the ends of the complementary DNA strands. That's why it is called invertron. Okay. So telomere replication or self-renewal. How exactly this telomere replication happens, and the self-renewal in the sense, since this particular lagging strand or all the other things that we will discuss will come to know. So telomere replication is an important aspect in DNA replication. Without telomere replication, there is no DNA replications, as the telomeres are required for the DNA replications, not only DNA but chromosome replications and stability also. So the primary difficulty with the telomeres is the replication of lagging strand. So in telomeres what happens? The replication of lagging strand will be missed out. So that means the last part of the last end of the strand, okay, that will be missed because of some reason. Now what is that reason? That we will see. So because the DNA synthesis requires RNA template, agree. So without DNA RNA template, there is no DNA synthesis. That is we know transcription has to be there. That means for reverse transcription, if you are talking about, right, reverse transcription has to be there to create DNA, or even if you are simply talking about this thing, central dogma, like a reverse transcription. So because DNA synthesis requires the RNA template, so that provides a free hydroxyl group, right? 3' OH group should be there, which will provide RNA template which is necessary to prime or to start the DNA replication. So and this template is eventually degraded. That we know that RNA template is there, no? That usually eventually it degrades over the period of time. So a short single stranded region would be left at the end of the chromosome. When the RNA template degrades over a period of time, a short single stranded region will be remaining at the end of the chromosome. So that itself will act as a lagging strand. That means it is not yet completely replicated. Understanding? Why? Because in order to complete the DNA replications, we need RNA template. And here what is happening? RNA template is degrading. Once that is degrades, what happens? The short single strand region would be left at the end of the chromosome. So that has to be replicated, right? Understanding? So this region would be susceptible to enzymes that degrade single strand DNA, right? So any enzymes that can degrade the single strand DNA, this particular region will be susceptible. So that can degrade that particular single stranded DNA. Now the result would be that the length of the chromosome would be shortened after each division. So every division, if RNA templates keep on getting degraded and lagging strand is forming, and next the same thing is happening. The same thing is happening in every the cycles or every division of chromosome. What happens? The lagging strand will keep on increasing, or the chromosome will become shortened. Why? Because the last part of the end part of the chromosome is being missed out. So now the action of the telomerase enzymes ensure that. Now that's why we need telomerase enzyme. Why? This telomerase enzyme ensures that the end of the lagging strands are replicated correctly. Understanding? So here the whole concept is revolving around the end of the chromosome should be protected, and it should be made sure that there is a replication from this particular end also. For that, we need telomerase enzymes. Understanding? So if and this, why the end of the chromosome or lagging strand is forming? Because the RNA template is degrading soon before the complete replications. Okay, so in order to get that, so if this continues, what happens? We will keep on getting shorter and shorter and shorter chromosomes in every division. Understanding? So for that, to save this particular the lagging strand, we should use the telomerase enzymes. Those enzymes can ensure that the ends of the lagging strands are replicated correctly. So let's discuss about telomerase enzyme. I hope you understood the introduction and why do we need telomerase enzyme. So telomerase was discovered in 1984 by Elizabeth Blackburn and Carol Greider of University of California in Berkeley. So it is a reverse transcriptase that synthesizes DNA using RNA template. So that is what we need, right? DNA should be there and with the help of RNA template. So telomerase enzyme is what? It's a reverse transcriptase enzyme. Okay, that synthesizes what? DNA by using what? RNA template. So unlike most reverse transcriptases or transcriptase enzymes, the enzyme itself contains the RNA that serves as its template, but here the telomerase can add new repeat units to the 3' end of lagging strand. So here we know that the enzymes, whatever the reverse transcriptases are there, they will have their RNA in their own template, but along with that, here this particular telomerase, it will add new repeat units. So that is the speciality of this enzyme. That's why it is called as telomerase. Okay, so that means it is adding the new repeated units, okay, to the where 3' end of the lagging strand, so that the lagging strand has to be replicated. Now, well-studied system involves the tetrahymena protozoan organisms. Okay, here the telomerase of this organism and in the sequence is 5' to 3'. We are having TT4G. So 2 times T, 4 times G. Okay, so 5' T, T, G, 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 3'. Okay, now the telomerase adds a series of this particular sequence, that is 5 to 3', T, T, G, G, G repeats to the end of the lagging strand. So once it is ending, that's why that's what we discussed now. This particular enzyme has a special character that adds a new repeat units, right? So that new repeat unit is repeated unit is what? TTGGGG. In well described protozoan organism we are taking as an example. Now the telomerase adds the series of somas, this particular sequence which repeats the end of the lagging strand. Okay. Now the hairpin occurs when the unusual base pair between the guanine residues in the repeat form. So obviously, because since when it is added, since it is unusual base pair which is coming in between the guanine residues in the repeat form. Now next what happens? The RNA primer is removed, okay, and the 5' end of the lagging strand can be used for DNA synthesis. This is the way it looks like. So here this is the telomerase enzyme, here it comes, and this is the hairpin kind of structure. We will be removing the RNA and 5' end will come into picture, and this is how it goes. And this particular telomerase enzyme, as discussed here, telomerase can add a new repeats units to the 3' end of the lagging strand. Now this is a lagging strand which is not completely transcribed, so it will add this particular sequence, that is TT4G, GGGGA to the 3' end, and complementary to that there will be a DNA synthesis. Okay, so DNA will be synthesized with the help of RNA template. So that is the function of telomerase. That's how it starts adding on. So once it adds, it brings like this, then it adds, that was for elongation, then the translocation will be there. That means this will move the other side, and the next set of the repeated unit will come, and that's how the lagging strand will be replicated. Okay, so the next RNA is removed, the 5' end of the lagging strand can be used for the DNA synthesis, for this particular DNA synthesis. That is, the lagging strand can be used for DNA synthesis. This strand can be used for DNA synthesis. Now the ligation occurs between the finished lagging strand and the hairpin. So this is the hairpin and this is the finished lagging strand. So ligation happens, that means the ligation will be there, joints will be there. So finally hairpin is removed at the 5 prime end of the at the 5 to 3 repeats. So here it is. So this will be removed, so this will come out. Okay, so and the process continues. Now the gap in the complementary strand is filled by the replication enzymes polymerase alpha primase. So that's how the gap will be filled. Okay, so the things that's how the things works. Okay, so that's how we can completely replicate the lagging strand also, that is with the help of telomerase enzyme. So that's it about the telomers and their the self renewal, and I guess that's it. And also in this topic we are having telomerase enzyme. So this can be asked for 7 marks or 8 marks, and also can be asked the telomerase itself as for 5 marks. Okay, so that's it. In the next topic we are having the cell migration, that we will discuss in the next class.